Everyone knows the Vikings reached North America before Columbus. This is not a fringe theory. It is not a romantic legend. It is an archaeological fact, confirmed since 1960, when Norwegian archaeologists Helga and Anne Stein Ingstad excavated a Norse settlement at Lonceau Meadows, in Newfoundland, Canada, dating to approximately 1000 CE. The Norse sagas had described Vinland for centuries. The archaeology confirmed it. The debate about whether the Norse reached North America was settled half a century ago. The questions that remained were harder and stranger. Not whether they came, but what happened when they arrived. What happened when a culture of Scandinavian seafarers, already carrying the genetic legacy of the Viking expansion across Europe and the North Atlantic, encountered the indigenous peoples of North America? Did they mix? Did they trade? Did they fight and leave? Or fight and stay? Did any of the Norse settlers remain long enough to have children with the people they called Skraelings, the indigenous inhabitants of the lands they were exploring? And perhaps most unexpectedly, did any of those encounters leave a genetic trace that has persisted into the modern world? In 2021, two separate ancient DNA studies addressed these questions from different angles, and the findings that emerged were, in combination, more surprising than either study alone would have suggested. The first study, published in the journal Nature in 2021, was not primarily about the Norse at all. It was a comprehensive genomic study of ancient indigenous North American populations, conducted by a large international team. But embedded within its findings was something that no one had specifically set out to look for. Among the ancient individuals analyzed, a small number from the northeastern North American region showed a genetic component that appeared to derive from a European source, specifically, a source consistent with Norse or early medieval Scandinavian ancestry. The individuals in question were from a period and a location that made post-Columbian European contact an unlikely explanation. The signal was not large, it was not present in all northeastern populations, and the researchers were careful to note that it required further investigation before firm conclusions could be drawn. But it was there, consistent across multiple individuals, and it pointed toward a contact event between Norse settlers and indigenous North American populations that was not simply the brief commercial exchange described in the sagas, but something more intimate and more biologically consequential. The second study, published later in 2021 in Science Advances, took a different approach. Rather than looking at indigenous populations for Norse ancestry, it looked at modern Icelanders and Greenlanders for indigenous North American ancestry. The Norse colonization of Greenland beginning around 985 CE is well documented. The Greenlandic Norse settlements lasted for roughly 5 centuries before collapsing around 1400 CE, for reasons that are still debated. During those 5 centuries, the Norse Greenlanders had repeated contact with the indigenous Dorset and later Thule people of Greenland and the Canadian Arctic. What the Science Advances study found was that a specific mitochondrial haplogroup, a maternal lineage marker designated C1e, which is otherwise found only in indigenous North American and Greenlandic populations, appears in the genealogies of modern Icelanders at a low but statistically detectable frequency. The haplogroup traces back in Icelandic genealogical records to the early medieval period. Its most likely explanation, the study's authors concluded, is that an indigenous North American woman, or a woman descended from indigenous North American ancestry through the Norse-Greenlandic contacts, traveled to Iceland during the period of Norse North American exploration and settlement. She was there. The DNA proves it. A woman from the indigenous world of North America arrived in Iceland sometime around 1000 CE, had children, and her maternal lineage has persisted in the Icelandic population for over 1,000 years. The Norse sagas do describe interactions with the Skraelings that go beyond simple trade. The Saga of the Greenlanders and Eric the Red Saga both describe periods of coexistence and exchange that occasionally included the acquisition of indigenous people, whether by trade, capture, or voluntary accompaniment is unclear. There is at least one saga reference to a Skraeling boy named Vifil who came to Iceland. There may have been women whose stories the sagas did not record at all, whose presence in the Norse world was unremarkable enough or unimportant enough to the saga writers that they were not mentioned, but whose maternal genetic contribution has persisted through 40 generations of Icelandic mothers and daughters. The scale of this contact should not be overstated. The Norse presence in North America was brief. The the Vinland settlements lasting perhaps a decade or two at Lanso Meadows before being abandoned, probably due to conflict with indigenous populations who were numerous enough to make permanent Norse settlement untenable. The genetic signal in modern Icelanders is small. The signal in ancient indigenous North Americans, while present in the 2021 Nature study, requires additional confirmation and is not yet the consensus finding it may eventually become. But the direction of the evidence is consistent. Contact happened. Some of that contact was intimate enough to produce children. Some of those children's descendants are living today in Iceland, carrying in their mitochondrial DNA the genetic record of a woman who crossed the Atlantic from North America to Europe 1,000 years before the contact that history recognizes as the beginning of the transatlantic connection. The Norse in North America story has a further layer that the DNA studies illuminate, one that concerns the Norse themselves rather than their interactions with indigenous people. The population that sailed to Vinland was not a simple Viking raiding party from Norway. By the time of the Vinland voyages, the Norse expansion had already transformed the genetic landscape of the North Atlantic. The settlers of Iceland in the late 9th and 10th centuries were predominantly of Norwegian origin, but included significant numbers of individuals from the British Isles, particularly from Ireland, Scotland, and the Northern Isles, populations that the Norse had conquered, settled among, traded with, and intermarried with across generations. The ancient DNA of the Viking Age, studied comprehensively in a landmark 2020 paper in Nature that analyzed 442 ancient Viking-era individuals from across Scandinavia and the Norse diaspora, showed that the Viking world was genetically diverse in ways that the popular image of blonde Scandinavian warriors does not capture. Norse individuals from the British Isles settlements showed substantial Celtic ancestry. Norse individuals from the Russian river networks showed Eastern European and Central Asian admixture. The people who sailed to Greenland and from Greenland to North America were carrying within them the accumulated genetic heritage of the Norse expansion, which was itself a mosaic of Scandinavian, Celtic, and various other European ancestries. When those people met the indigenous populations of North America, it was not a meeting of two pure and separate genetic streams. It was a meeting of two complex, historically accumulated populations, each the product of migration, mixing, and the ordinary processes of human genetic accumulation. The Norse were already a genetically diverse people when they arrived. The indigenous populations they encountered were equally, if differently, complex. The contact between them, brief as it was, added yet another layer to the already complicated genetic history of both populations. The Lanso Meadows site in Newfoundland remains the only confirmed Norse archaeological site in North America, but the 2021 studies, combined with the broader 2020 Viking Age DNA study, suggest that the Norse presence in North America was not simply the brief episode of failed colonization that the archaeological record at Lanso Meadows describes. There were multiple voyages. There were extended periods of contact with indigenous populations in Greenland, and possibly in the Canadian Arctic and subarctic. There were people, almost certainly including women, who moved in both directions across the North Atlantic. The sagas preserve some of this. The saga of the Greenlanders names Gudrid Thorbjarnardóttir as a woman who made multiple transatlantic voyages, gave birth to a son named Snorri in Vinland, who was therefore the first European child born in North America, and eventually traveled to Rome as a pilgrim. Her story is one of the most extraordinary in the medieval world. But there were other women whose stories the sagas did not tell, whose journeys were not recorded because the saga writers were interested in the adventures of men, and whose presence in both worlds is now visible only in the genome. The indigenous woman whose C1e haplogroup appears in Icelandic genealogies was one of those unrecorded women. She had no saga. Her name is unknown. The circumstances of her journey to Iceland, whether she chose it, was taken, or followed a partner she had chosen, we cannot know. But she was there, and she is still there, in a molecular sense, in the mitochondrial DNA of Icelanders, who have no idea they carry the genetic legacy of a North American indigenous woman who crossed the Atlantic 1,000 years ago. What does the Norse in America story ultimately tell us about human prehistory and about the nature of the transatlantic connection? First, it tells us that the Norse contact with indigenous North America was not the sterile, brief, failed encounter that a simple reading of the archaeological evidence at Lanso Meadows might suggest. It was extended enough, over the full arc of Norse-Greenlandic contact with indigenous peoples to produce biological consequences that are still detectable in modern populations on both sides of the Atlantic. Second, it tells us that the transatlantic connection, in the genomic sense, predates Columbus by at least 500 years. Not in a way that transformed either population demographically, not in a way that established lasting cultural exchange, but in a way that left a real molecular trace. The first transatlantic genetic exchange was not European colonialism. It was a Norse woman in North America and an indigenous woman in Iceland, or some combination of such encounters, around 1000 CE. Third, it raises the question that the genetics cannot yet fully answer. How many such encounters were there? How many individuals crossed the North Atlantic during the Norse period of contact, in both directions, and left genetic traces that we have not yet had the sensitivity or the sampling strategy to detect? The Icelandic population is among the most thoroughly genetically mapped in the world, which is why the C1e haplogroup was detectable there. Most other populations that were part of the Norse contact network have not been studied with the same resolution. The Norse-Greenlandic settlement lasted 5 centuries. 5 centuries is a long time for people to have children, and for those children to have children, and for genetic legacies to travel in ways that no saga recorded. The Norse in North America were never what we thought, because what we thought was that they came, saw, failed, and left without consequence. The DNA says they came, made contact that was intimate enough to produce children, and left a molecular trace that has persisted for 1,000 years on each side of the ocean. They failed to colonize. They did not fail to connect. And the connection they made, brief and inadvertent and unrecorded as it was, is written in the genome of people who have been living with it for 40 generations without knowing it was there. There is one more thing the Norse in America story illuminates that goes beyond the specific question of Norse indigenous contact. It is a reminder of how much of the human past is invisible to conventional history. The saga writers were literate, sophisticated, attentive to the details of voyages and battles and political alliances. They preserved a remarkable record of Norse life in the medieval North Atlantic. And yet they missed something as fundamental as a woman crossing the Atlantic from North America to Europe and establishing a genetic lineage that has persisted for 1,000 years. What they missed because they were not looking for it. The categories through which they understood and recorded the world did not include the idea that a Skraeling woman's children would matter to the genealogical record of Iceland. Her children were, from the saga writer's perspective, Norse children. Their mother's origin was not the story. The genome makes her origin the story. It recovers what the saga overlooked, and in doing so, it demonstrates something that every ancient DNA study in this series has demonstrated in different ways. The written record of the human past is incomplete, in ways that no amount of careful reading can fully reveal, because the incompleteness is structural. The record was made by people who were interested in certain things and not in others, who understood the world through categories that made some people visible and others invisible, who recorded some crossings and overlooked others. The DNA records all of them. Every crossing, every child, every woman whose name the saga writers did not think to write down. The genome is the history that no one chose to leave behind, and no one could choose to destroy. It is simply there, in the cells, waiting for the science that could finally read it. And when we read it, we find a world that is always larger, always more connected, always more human than the histories we wrote about it managed to capture.